Well, hey AP, hope you're doing well when you get this video. As promised, here are the Transpiration Lab expectations as far as the report goes. I hope this video helps. Of course, all this will be under topics on the Transpiration Lab topic. But without further ado, let's begin, okay? So I wanted to discuss with you first about expectations in general. So this is the general flow of a lab report. I'm not going to throw all this at you first go around. That would be cruel. But I do want to highlight a few aspects, okay? Definitely a title, concise, explains the purpose of the investigation. We're skipping abstract in this lab, but I would read that over. Introduction, of course, background information, talking about transpiration, water potential possibly, stomata, osmosis. All of that would be valuable information. Purpose, of course, why are we doing what we're doing? That's always helpful to see. In a lab report, hypothesis, if blah, 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 then blah, blah, blah. Practice writing that with your partner, of course. We will work on null hypothesis writing, but not for this lab. Materials, just a list. We're not going to worry about procedures. Data is going to be huge in this lab. I'll show you exactly in a second what I'm expecting there. And of course, conclusions and discussions. We're going to skip the last two. Not the third one. Grammar, punctuation, spelling, always in school, right? But results summarized, errors identified. Use data though, right? We're using our data to come up with results, come up with conclusions. Was your hypothesis rejected or accepted? What did you learn? All of that's based upon data. Remember, data is telling a story. A few other notes. Lab should be written in pen, blue or black. You know, we want this work to be permanent. We want to see how we have grown throughout the year. Pencil can get messy. Pencil can be erased. We don't want that. I saw this online reading through lab reports, guidelines from universities, but fluorescent pink, electric green, they're hard to read, a little annoying in the world of lab reports. I actually had a former student who wrote in pink for tests sometimes, and they weren't crazy hard to read, but it was only multiple choice. I can see how reading lab reports in fluorescent pink might cause a headache. Labs should be legible. Illegible notebooks will be penalized. After title, please put your partner's name. Hypothesis, no use of slang, not slang, I don't even know what that is. No use of slang, complete sentences, if-then statement, okay? Pre-lab discussion questions, you can put that in the introduction. Procedure, the only thing I would like for you to put there is what was your independent variable and what was your dependent variable. Okay, we don't have to worry about this one right now, but definitely independent variable and dependent variables are identified. Data, use a straight edge ruler when you're making tables or graphs. That's helpful. We don't want this to be messy. We want to do good work. Make it at least half a page. Yeah, data, since it's telling a story, let's make it large, right? Let's make this story large. So drawings, there will be a few drawings. If you can draw something and explain it, you know it pretty well. So there will be a few drawings, title the drawings. You may use a pencil during this. And then the conclusion, a short paragraph, of course, restating the hypothesis. Do you support or reject it? Again, we're talking about data here. We're talking about, does the data support or refute the hypothesis? Are we accepting it? Are we rejecting it? Do we need more experimentation? What is going on? And then finally, include post-lab questions that would be important to see in the conclusion as well. Use third person during lab reports. No I, we, us, you. Third person would be great. So let's review just a little bit. This is transpiration. Okay, so it's all about water potential. Going from high to low. Osmosis goes from high freedom, lots of water, to low freedom of water. And remember the cells that we talked about in class, the hypertonic, hypotonic, isotonic solutions in blood. Okay, so what happens there in the roots? Negative water potential draws water into the roots. So lots of water in the ground. This is of course a tree, way, a lot taller than our coleus plants. But cohesion and adhesion, of course, is properties of water that draw that water up. So it's kind of like in this long chain. And so transpiration draws water from the leaf. So literally water is leaving as water vapor through the leaf, and that's through one stoma. So there's different layers. This is the xylem. And so as you can see, there's this potential gradient. So this is what keeps water, this is the gradient that drives water up that tree. Okay, so it's facilitated by osmosis, root pressure, and the physical chemical properties of water. Here is, oh man, that doesn't look good. Here is the stoma opening. So as you can see, lots of water around, stoma is open. When not much water around, stoma is, the closing. Might differ, depending upon the situations. Here's an electron picture about, microscope picture about open and closed stomata. These are the objectives of the labs. I'm not going to read these, but you can pause the video if you like to go back and read them. But I do want to read the 3 that are in your student manual. To investigate the relationship among leaf surface area, number of stomata, and the rate of transpiration. To design and conduct an experiment. Again, you were doing that in AP Biology. To explore other factors, including different environmental variables on the rate of transpiration. And then finally, to investigate the relationship between the structure of vascular tissues and their functions in transporting water and nutrients. That will be mostly in the conclusion in your lab report. So, pre-lab questions. This will be a drawing. So if a plant cell has lower water potential than its surrounding environment, make a prediction about the movement of water across the cell membrane. In other words, will a cell gain water or lose water? Explain your answer in the form of a diagram, so draw it with notation. You can label it, draw arrows. What is happening in the drawing? Pre-lab question number 2. In the winter, salt is sometimes spread over icy roads. In the spring, after the ice is melted, grass often dies near these roads. What is happening? Again, diagram this. This is an art class, right? Science class, but diagram this with annotations. Describe, label what is happening. Briefly describe the difference between xylem and phloem. Sorry, it's going to be in pre-lab questions, but I do want you to address it maybe a little bit in conclusion. Briefly describe this. So maybe a little research here. Again, I'm not looking for many sentences. 3 to 5 sentences would be fine with that. So the hypothesis. Begin thinking about a null hypothesis. You can look that up if you're not sure what that is. We will talk about it more as we go. But write hypothesis with your partner if you haven't done so already. If-then statement. So data, the leaf surface area. The actual drawing of leaf on graph paper, which we did in class, that would be amazing to put in your lab notebook. Cutting it out and gluing is fine. Hopefully you kept it. If you didn't, I would come talk to me. We'll find out what to do. And so here's the data. This picture is on topics. This is our data that you can see. You'll probably have to zoom in for a few of them. I think they're all, legible. I think you can read them all. I might have to zoom in a little bit on a few, but this is the data table that we saw. Of course, we missed a few days due to the weekend and then block day, but we did our best. So we can see that data table right there. And so as far as the data goes, the data section that is, I want a table that shows the measurements for each day, including percent change. And so I gave you a little tip here. I want this handwritten. Lab reports are handwritten. It helps us learn. It triggers different parts of the minds, instead of letting a computer do it for you. The formula, though, there is a formula, and there is an Excel way to kind of help manage time a little bit that I want to show you. Graph, I want you to illustrate this data, right? Data tells a story. Shows percent change for each variable over 7 days. So there are correct ways and wrong ways of illustrating this data. I'm not going to tell you those right now because I want you to use your resources, talk with your partners, figure out how you should visually represent the data that you see. But let me go to Excel right now. And so this is just an example, and there's many different ways you could probably do this table, but this is just what I was kind of doing. So I started with wind. I believe that's just the top one on the board, but you can see day 0, okay, initial readings 364.9, 390.4. This is in grams. You will need that labeled on the graph. And so control, day 1, 351, 375. Okay. So what's happening though, right? And so we can see our percent change here. So percent change, negative 0.037. Again, we can make that a percent, 3.7%. And then by timesing by 100, this is decimal form, and then negative 0.03868. So how do you do this? So what you do, in Excel at least, Excel has all these formulas built in. Excel is actually an amazing, amazing program. And just to manage some time, you could do this if it will help you. So what I do, and percent change of course, is new minus old divided by old. So new, and here's the thing, you need parentheses though. So parentheses, new minus old, original, initial, divided by initial. Okay, press enter. And so we're at like 10.8%. That's like something definitely is happening. And then you can do it over here too. So equals parentheses new minus old, close parentheses, divided by old. And you can see that the wind, at least at the beginning of the experiment, losing a little bit more. We could do some statistical analysis to figure out if this is meaningful data or not. We will be doing that eventually, but not right now. Again, first lab report, let's begin at the beginning. And so, but that is a helpful way to do that. So I do want a table. Again, let's review. I do want a table that shows measurements for each day, including percent change. And then I want a graph that correctly illustrates the percent change for each variable over 7 days. Okay? And then finally, the last part, draw conclusions. So I want you to summarize the experiment, 3 to 5 sentences. Discuss the results that you found and how this relates to transpiration, water potential, and stomata. Draw relationships here. Draw conclusions based upon data. Accept or reject your hypothesis. Based on data, which variable resulted in the greatest percentage of water loss and why? Again, why is important in conclusions. Investigate how guard cells, this is a question in your lab manual, investigate how guard cells control the opening and closing of stomata, including the role of acetic acid and potassium. And then finally, any errors or anything you would change, and where could you go from here? Again, a few sentences on the last part. But this is the Transpiration Lab. Very, kind of straightforward lab, but, definitely great practice in scientific method, data collection. And then, of course, we're going to practice writing lab reports. So again, this is due. Make sure you check plan book on when it exactly is due. But I hope this was helpful.